Hello students, welcome in this online video lecture and once again in this second semester M.Sc. part for students I am going to teach you paper CCO 250, that is photocyclic chemistry, pericyclic chemistry and spectroscopy. I hope that all of you are safe, healthy and follow the rules and regulation. M.Sc. पार्ट फर्स्टच्या सगळ्या विद्यार्थ्यांचं मी या ठिकाणी द्वितीय सत्रामध्ये स्वागत करतो. मित्र हो, कोरोना पॅन्डेमिकच्या पार्श्वभूमीवर तुम्ही सगळे सेफ आहात असं गृहीत धरून आपण छानपैकी आपलं दुसरं सेमिस्टर या ठिकाणी सुरू करतोय. And in this semester I am going to teach you the paper CH-250 organic chemistry. Total title of this paper is photocyclic chemistry, pericyclic chemistry and spectroscopy. This course includes these three particular main topics in two sections, that is photochemistry and pericyclic reaction we are going to study in second half. In this first half we are going to start section spectroscopy, and this spectroscopy we have to study the four different spectroscopy, that is UV visible spectroscopy, infrared spectroscopy, NMR spectroscopy and mass spectroscopy. According to distribution and this situation, forget about the atmosphere of outside. हेल्दी राहा, रिलॅक्स राहा. ऑनलाईन लेक्चर मधून जेवढं तुम्हाला हेल्दी ठेवता येईल तेवढं छानपैकी लेक्चर मधून तुम्हाला मी या ठिकाणी स्पेक्ट्रोस्कोपी प्रोव्हाइड करतो. ऑल दिस टॉपिक in details, spectroscopy related topics you have studied in third year B.Sc. 20% part only additional at this M.Sc. level. Okay, so what is the distribution? Technically I provide video lecture and later on, if there is no any technical problem, I am going to most of the topics in online form. Okay, so don't forget about the topic and steps. In this lecture we are going to start our first spectroscopic method or analysis, that is ultraviolet or ultraviolet visible spectroscopy. In short it is called as UV spectroscopy. This is the first type of spectroscopy, that is UV spectroscopy or ultraviolet visible spectroscopy. Okay, now this type of spectroscopy is also known as इलेक्ट्रॉनिक स्पेक्ट्रोस्कोपी. UV स्पेक्ट्रोस्कोपीलाच आपण दुसऱ्या कुठल्या नावाने म्हणू शकतो, तर दिस इज इलेक्ट्रॉनिक स्पेक्ट्रोस्कोपी and it is used to measure. What is the use of UV spectroscopy? This spectroscopy is used to measure the number of conjugation of double bond and aromatic conjugation present within the molecule. Okay, व्हिडिओ लेक्चर ज्यावेळेस तुम्ही वॉच करताय. समोर तुमच्या समोर व्यवस्थित एक स्लाइड आहे आणि मी ऑडिओमेट्रिकली तुम्हाला ते डिस्कस करतोय. ओके. तर UV spectroscopy means electronic spectroscopy. What is the use of this ultraviolet spectroscopy? That is, this spectroscopy is used to find out the number of conjugation of double bond and aromatic conjugation present within the molecule. Is it right? First we say, what is conjugation of double bond? Is it right? Whenever any molecule contains double bonded system in alternate manner, एक डबल बॉन्ड आहे, एक सिंगल बॉन्ड आहे, परत एक डबल बॉन्ड आहे, त्या सिस्टीमला आपण म्हणतो कॉन्जुगेटेड सिस्टीम. फॉर एक्झाम्पल, ethane is a simple double bonded system. It does not contain multiple system, means it is having lack of conjugation. Second thing, benzene contains three double bonds which are having in alternate manner, means it is containing three double bonds in conjugation with each other. So first important principle of UV spectroscopy is to determine the number of conjugation present in the system. Okay, first we have learned that is those organic molecules, that is aldehydes, ketones, dienes and polyalkenes, which are containing conjugated system, and such type of conjugated system whenever present, this whether it is present or absent, it can be determined by ultraviolet visible spectroscopy. Is it right? Clear? Okay. Second thing, how it works. How it works. So, UV spectroscopy always involves the excitation of electrons from HOMO, that is highly occupied molecular orbital, that is ground state energy level, to lowest unoccupied molecular orbital, that is excited state. Means by using ultraviolet visible light, whenever any compound is exposed to UV light radiation, UV light radiation मध्ये कोणत्याही प्रकारचा मॉलिक्यूल, ऑर्गॅनिक मॉलिक्यूल which is active, whenever it is exposed to ultraviolet radiation, then there occur changes in electronic energy level. Understand? That is, in electronic energy level, electrons are get excited from highly occupied molecular orbital, that is ground state energy level, to lowest unoccupied molecular orbital, that is excited state energy level, so that there occur change in transition, change in energy, and hence the promotion of electron takes place in ultraviolet spectroscopy. The measurement include change in electronic energy transition level, so that the organic molecule shows such type of changes. Is it clear? Again, I will repeat how it works, which changes are observed. That is, in case of ultraviolet visible spectroscopy, whenever any organic molecule is exposed to UV light radiation, it absorbs the light radiation. Due to absorption of light radiation, electrons from ground state are get excited to excited stage, and this change in energy level is measured in terms of energy absorbed. This is the basic principle of UV spectroscopy. Now, how this energy is measured? UV spectroscopy, that is the ultraviolet region or UV visible light radiation, always having the measurement of light radiation in terms of nanometers. कशामध्ये मोजलं जातं? नॅनोमीटर्स मध्ये. and range is from 200 nanometers to 800 nanometers. 200 नॅनोमीटर पासून 800 नॅनोमीटर पर्यंत जर रेंज असेल त्या लाईट रेडिएशनच्या एनर्जीची, तर त्या ठिकाणी ती जी लाईट रेडिएशन आहे त्याला आपण म्हणतो अल्ट्रा व्हायलेट लाईट रेडिएशन. Again further it is divided into two regions, that is ultraviolet visible spectroscopy. Ultraviolet region corresponds to 200 nanometers to 400 nanometers, while visible region is corresponds to 400 nanometers to 800 nanometers. कुठल्या टाईपची लाईट? की ज्या लाईटची फ्रिक्वेन्सी रेंज ही 200 ते 400 UV रिजन येतो त्याला म्हणायचं, आणि त्याची रेंज 800 पासून 400 नॅनोमीटर किंवा 400 नॅनोमीटर पासून 800 नॅनोमीटर अशी असेल, तर ह्या टाईपच्या रिजनमध्ये येणाऱ्या लाईटला आपण UV लाईट रेडिएशन्स म्हणतो. सेकंड थिंग, अगेन आय विल रिपीट, पुन्हा मी रिपीट करतो की UV लाईट रेडिएशनमध्ये लाईटची वेवलेंथ कशात मोजली जाते? तर नॅनोमीटरमध्ये. रेंज काय आहे? 200 ते 400 नॅनोमीटर इज द UV region. Visible region is 400 to 800 नॅनोमीटर्स. आणि टोटल रेंज सांगायची असेल तर 200 नॅनोमीटर पासून 800 नॅनोमीटर हा जो रिजन आहे, this region is the UV spectroscopic light radiation region. Okay, means now again I will repeat what is the basic principle. Those organic molecule containing double bond system or multiple bond system, that means it requires presence of conjugation. Whenever it is exposed to UV light radiation, it absorbs some of the light radiation in the region of 200 nanometers to 800 nanometers. Change in electronic energy level takes place, that is electrons are excited from ground state, त्यालाच आपण म्हणतो HOMO, HOMO, to excited state energy level, that is LUMO. And how much energy is absorbed, this decide the presence of conjugated system presence in this particularly double bonded system. And hence, according to UV spectroscopy, we can measure the number of conjugated system. The gap between HOMO-LUMO decreases as the conjugation goes on increasing. Okay, next one. What is the base of this particular UV spectroscopy? UV spectroscopy is totally based on Beer-Lambert's law. All of us are familiar with Beer and Lambert's law. That is, UV spectroscopy, how it is depends on Beer-Lambert's law. Whenever, what is the Beer and Lambert's law? That is logarithm of intensity of incident light I0 upon intensity of transmitted light. आता I0 upon I हा जो रेशो आहे, incident light means what? That is UV light radiation, how much intensity it provide from its source, त्या लाईटला आपण काय म्हणतो? incident light. And after transition of electronic level, some of the light radiations are get transmitted. What is the pattern? Some light radiations are absorbed, some are transmitted, and some are reflected. But in this case, we are considering how much light is provided, that is the intensity of incident light radiation, how much amount of light radiation is absorbed, and how much light radiation is transmitted. That's why logarithm of I0 upon I, that is intensity of incident light radiation divided by intensity of transmitted light radiation, is equal to molar absolute absorptivity into path length of the sample into concentration of sample is equal to absorbance divided by concentration into particularly path length, that is absorptivity कशी काढली जाते ते पुढे सांगितले molar absorptivity. But the total UV visible spectroscopy is totally depends on Beard-Lambert's law. सगळ्यांना Beard-Lambert's law माहिती, that is logarithm of intensity of incident light radiation divided by intensity of transmitted light radiation is equal to molar absorptivity into path length of that particular path or cuvette into concentration of solution. And according to this, we have to find out how much amount of light radiation is transmitted. From this amount of light radiation transmitted, we can find out the how much light radiations are absorbed, किती light radiation absorb केले हे find out करू शकतो. And from this absorbance we can find out the how much absorbance is directly proportional to number of conjugations present in that double bonded system of compound. Hence absorbance we have to find out, and according to Beard-Lambert's law, absorbance is the reciprocal of transmittance. Transmittance means transmittance can be calculated from intensity of transmitted light. Is it right? So to calculate absorbance, absorbance capital A is nothing but it is a reciprocal of transmittance. Absorbance is also called as optical density capital D, and absorbance is equal to logarithm of reciprocal of transmittance or the log of I0 upon I. Means by using Lambert-Beard's law we can find out initially transmittance. From this transmittance we can find out optical density, and optical density is nothing but it is a transmittance. So here you have to remember two major terms. First, Beer-Lambert's law. Second, absorbance. Absorbance कशाला म्हणतो आपण? The absorbance is nothing but it is the reciprocal of transmittance or the concentration or intensity of light radiation which is absorbed by substance, or in this case by the particular organic molecule. त्याला आपण काय म्हणतो? Absorbance. Now how this absorbance is carried out? Absorbance is carried out by using the measurement of transmittance, and transmittance, that is absorbance is equal to logarithm of 1 upon T or logarithm of I0 upon I. The next term comes, that is the what is transmittance. Transmittance is nothing but the fraction of incident light radiation transmitted from organic molecule is known as transmittance. How much fraction? Incident light मधला किती फ्रॅक्शन ट्रान्समिट झाला एक्सपेरिमेंट करत असताना, तर ट्रान्समिट झालेला जो फ्रॅक्शन आहे तर त्याला आपण म्हणू शकतो ट्रान्समिटन्स. सो दिस इज अबाउट द टू इम्पॉर्टंट टर्म्स, absorbance and transmittance. In UV spectroscopy technique we are going to measure the absorbance, and absorbance can be calculated from transmittance. Okay, we are going to stop here in this first half of this lecture. Okay.